Okay, so in this video we're going to go over Lab 11 preview of gene expression. So as we know, every cell contains all the DNA in our genome. Some cells have this gene off. Certain genes are off in certain cells, while others are on in certain cells, and they transcribe into mRNA. So we can measure this with the assay we're going to do today. And this is important because cells don't want to have all their genes on at once. They want to save energy, and different cells need different things done. So obviously you don't want you don't want protein products that you need in your neurons to be firing off in your intestinal cells, for example. So the basic premise of this model that we're going to look at today is that there's a gene right here for, it's called beta, like galactose, I'm not going to spell it, but it breaks down lactose breakdown. So this will ultimately culminate with, protein over here that's going to break down lactose, but you don't want this gene to be on if there isn't any lactose around. So for the most part, although RNA polymerase, let's just call it this here, although this binds here, a lot of the times there is a negative repressor that's stuck on here. The only way to get the repressor out of the way so RNA polymerase can start transcription and make mRNA out of this DNA is that lactose itself has to bind to the repressor. And lactose will only show up if the bacteria is like in the presence of lactose. Otherwise, this gene stays off. But in the presence of lactose, we get rid of the repressor, and now we have mRNA for the lactose breakdown enzyme that's going to go and gobble up all these lactose. Okay. So in our case, what we're going to do is we have something called IPTG, and this is going to mimic the binding lactose to the repressor, because we're not actually going to add lactose. So that's our first mimic. The next thing we're going to add, then this is just like the breakdown first. ONPG. This is going to mimic the lactose that is going to be broken down by the large enzyme. And that's going to split it into galactose and ONP, which is from this. And the thing about ONP is that it will light up at 420 nanometers at a wavelength that we can measure, and it's going to show up as yellow. I don't have a yellow marker, sadly. So this product here from ONPG is going to be digested by the enzyme, if it's present, into a yellow reading that we can read at 420. So just give me a sec, I want to make sure I'm doing all these right. So tube 1, 2, 3, 4, 5, 6, 7. So for each of these tubes, you're going to add like our first thing, so it's half a mil of buffer, so 500 microliters buffer, 2 drops of chloroform into all these, and 1 drop of SDS to the tube. And this is going to serve as kind of the buffer for the reaction. Then for sample 1, so after you've added those first little chemicals in there to each one, sample 1, we're going to add bacteria cells. And these actually have the enzyme capable of beta-galactase. So we're going to add about 500 microliters of those cells. So at this stage, 2 minutes, oops, wait. Sorry. Yes, okay. So at this stage, so here's our bacteria in green, and this is tube 1. Okay. And then at this next stage, as the instructor, I'll add 3 mils, so quite a bit. No, that's the same color. I'll add 3 mils of the IPTG. And remember, this is our mimic that's gonna take the repressor off of that gene and allow transcription. So this is gonna be added and at time point 0. Then 2 minutes later, we're gonna add some of this mix in the tube that has IPG, bacteria, everything else. We're gonna add that and place about a half a mil of that from here into tube number 2. We're gonna do the same thing for 5 minutes here. 10, I think it's 15, and 30. And each successive one is gonna happen at these different time points. So that means that tube number 1 and tube number 2 have had the most time exposed to this IPG that's gonna get rid of the repressor and start gene transcription. So 7, on the other hand, is just, this is gonna be our blank. And actually all we're gonna add to this is just IPTG, I believe. Let me just make sure. Each tube, yeah. So this one, this is just our blank because we're never gonna add any bacteria to it, for example. Oops, wait. Yeah, because, sorry about that. So every other one has bacteria. 7 does not have bacteria. So you can just add a little green in there. You'll see this when you read it. And then after that, you'll kind of like put these in a heat block so that this reaction can happen. And then from that stage, the final thing that we're gonna add is that product. Remember, it's ONPG. And this is what's gonna mimic the breakdown of lactose, and this is gonna give us that yellow color. And we're gonna add this, just 0.2, to each tube at the same time, including 7, which won't make any color. So with this in mind, we can have like a string of time amounts after we've added the enzyme to activate the gene. Then we add in ONPG, which will get degraded. So we're gonna see, because everything should be active after adding this, we're gonna see at what time point the max gene expression has happened. And that should give us a clue about how quickly these things can turn on and off in response to very little stimulus from here.